Hi there, we're continuing with cell biology in a nutshell. Um, we left off learning about the way that phospholipid molecules form the membrane of cells and the way they pack based on their cylindrical shape. Phospholipids, shown here, they have a polar head and a nonpolar tail. The head consists of a glycerol backbone and a phosphate group modified by an alcohol group. So that's the hydrophilic or water-loving end of the molecule. And the other end is two fatty acid tails, as you can see. That's the hydrophobic side of the molecule, which repels water. So based on this shape, the molecule forms a bilayer where the non-polar fatty acid tails attract or group together in the inner part of the bilayer, while the hydrophilic polar head groups come on the opposite sides toward where the water is, and the polar side interacts with the water-soluble side of either the extracellular fluid or intracellular fluid. So we saw the structure of the molecule exactly, and usually it has one fatty acid tail next to another. One is usually just a straight chain, which is called saturated, and the other one is a kinked chain, which is called unsaturated, because it contains a double bond in the chain, causing a kink in the fatty acid tail. So this here shows the diagram 3D of how the molecule looks with the kink in it. So it's not perfectly linear, and this is the reason why the membrane is somewhat fluid and doesn't pack too closely, allowing it some kind of flexibility and movement of the membrane. So the phosphate group, which is a phosphate, 3 oxygens, attaches to the glycerol, and the modification of an alcohol polar head group as well is always part of the phospholipid molecule. In this case, it's choline. It could be different. So packing arrangements of lipid molecules in an aqueous solution occur spontaneously. Wedge-shaped lipid molecules form micelles, whereas cylindrical-shaped phospholipid molecules form bilayers. A lipid molecule and a lipid bilayer can be seen in such formation. Lipid molecules spontaneously form either one or other of these structures in water, depending on their shape. And well, as you can see, the wedge-shaped molecule has only one fatty acid chain coming out. The other one has two, which is normal, usual, 2 fatty acid tails. Being cylindrical, phospholipid molecules spontaneously form bilayers in aqueous solutions or environments. In this energetically most favorable arrangement, the hydrophilic heads face the water at each surface of the bilayer, while the hydrophobic tails are shielded from the water in the interior. The same forces that drive phospholipids to form bilayers also provide a self-healing property. A small tear in the bilayer creates a free edge with water. Because this is energetically unfavorable, the lipids spontaneously rearrange to eliminate the free edge. In eukaryotic plasma membranes, larger tears are repaired by the fusion of intracellular vesicles. The prohibition against free edges has a profound consequence. The only way for a bilayer to avoid having edges is by closing in on itself and forming a sealed compartment. So this is shown here. Energetically unfavorable is where you have a planar phospholipid bilayer with edges exposed to water, and it curves naturally, forming a sealed compartment. The phospholipid bilayer is in like a spherical shape, and this is more energetically favorable. The spontaneous closure of phospholipid bilayer forming a sealed compartment is more stable because it avoids the exposure of the hydrophobic hydrocarbon tails, fatty acid tails, to water, which would be energetically unfavorable. This remarkable behavior, fundamental to the creation of a living cell, follows directly from the shape and the amphipathic nature of the phospholipid bilayer. A lipid bilayer has other characteristics besides its self-sealing properties that make it an ideal structure for cell membranes. One of the most important of these is fluidity, which is crucial to many membrane functions. It was around 1970 researchers recognized that individual lipid molecules are able to diffuse freely within lipid bilayers. So various lipid molecules, for example, cholesterol, etc. The initial demonstration came from studies of synthetic lipid bilayers. Two types of preparations have been useful in such studies. One, bilayers made in the form of spherical vesicles called liposomes, shown here in a microscope, can vary in size from 25 nanometers to 1 micrometer in diameter, depending on how they're produced. Planar bilayers, called black membranes, form across the hole in a partition between two aqueous compartments. So here you have an electron micrograph of an unfixed, unstained phospholipid apparent and a drawing of a small spherical liposome, which is the same thing in cross-section. Liposomes are used as model membranes in experimental studies. So they're like vesicles. And then here you have another drawing. A cross-sectional view of a black membrane, a synthetic lipid bilayer. This planar bilayer appears black when it forms across a small hole in a partition separating two aqueous compartments. Black membranes are used to measure the permeability properties of synthetic membranes. Various techniques have been used to measure the motion of individual lipid molecules and their different parts. One can construct a lipid molecule, for example, whose polar head groups carry a spin label, quote unquote, such as a nitroxyl group, an NO, nitrogen and oxygen. This contains an unpaired electron whose spin creates a paramagnetic signal that can be detected by electron spin resonance, ESR spectroscopy. Electron spin resonance spectroscopy, which detects an unpaired electron, hence a paramagnetic signal. The principle of this technique are similar to those of nuclear magnetic resonance. The motion and orientation of a spin-labeled lipid in a bilayer can be deduced from the ESR spectrum. Such studies show that phospholipid molecules in synthetic bilayers very rarely migrate from the monolayer or leaflet on one side to that on the other. We'll continue in the next part. Thank you.